So this was about antibiotics production, and this is the same process for generalized as a generalized process depiction for the synthesis or the biosynthesis of antibiotics. Normally, we keep the spores or the cells in a freeze-dry condition. It's the best way we can have them because they won't be destroyed, but they won't be able to thrive. Then you culture them in agar slants. We would culture them into agar plates, agar tubes to a shake flask because it's very important so that they could start growing and dividing in small numbers. The seed is produced, and eventually from the seeds you could have them into these small vessels, and eventually they make all the way through the production fermenter. And from the production fermenter, you would have a filter, there will be a cooler, another filter. All these processes would ensure the maximum recovery of the product eventually to the downstream purification. Downstream purification is actually a technical word for the harvesting, cleaning, and purification of your products from biotechnological process using bacteria, mammalian cells, or plant cells, or fungal cells. Now, what are primary metabolites? How do they differ from secondary metabolites? The metabolism products, which are actually produced by organisms during their growth phase, so that they can be used for physiological functions, and they have a role in the overall support or development of the cell, are known as primary metabolites. For example, vitamins, for example, proteins, lipids, all these are primary metabolites. They have a direct role in, in supporting and performing physiological functions or structures of the cell. But when these primary metabolites are used and utilized by the bodies of organisms, then their end products could result into other products which are important in ecological activities. For example, in the competitions, for example, in case of antibiotics, all these antibiotics are released, or the bacteriostatic or the bacteriocins, they are released so that it kills the other competitors in the natural environment. And these are secondary metabolites. They do not have a direct role, or they do not have a direct necessity for performing physiological functions, supporting the cellular activities, and they do not have a role either in the structural or functional ways inside the cell. This is why the secondary metabolites are normally produced in the stationary phase, while in case of primary metabolites, they are in the growth phase, or let's say the dividing phase, which would be somewhere in between the log phase and the lag phase, mostly in the log phase where the bacteria are, or normal cells also, they are happily growing and dividing. The primary metabolites are, for this reason, produced in large quantities, and since they are produced in large quantities, it's easy to extract them. And in contrast to primary metabolites, secondary metabolites are produced in small quantities. This is why their extraction becomes difficult. The primary metabolites would be same in every species. For example, vitamins, carbohydrates, proteins, lipids, they are all the same in any kind of species of the world, either a plant or a microbe or an animal. But the secondary metabolites, species to species, they will vary. The importance of primary metabolites is like these products are used in industries for various purposes, for production, for a number of other purposes, and they play a very significant role in the cell growth, in the normal homeostasis, the reproduction, the development. Secondary metabolites are very important metabolites. For example, for us, they are very important metabolites because antibiotics are secondary metabolites, and antibiotics are the magic drugs or the magic bullets that keep us safe from a number of infections. Similarly, gibberellins, auxins, other hormones, they're also secondary metabolites, and they're also very important. They have a role in various activities. We discussed this in detail in genetic engineering as well as in plant tissue culture, the role of auxins and the gibberellins and cytokinins, how important they are for the generation of roots and shoots in tissue culture. And if we cannot have roots and shoots this differentiation in plant tissue cultures, then plant genetic engineering will be really, really difficult. Also involved in supporting and sustaining their life for long duration, as I told you, by wiping out the competitors out there in the ecological niche. Examples are for primary metabolites, vitamins, carbohydrates, proteins, lipids, amino acids, and for secondary metabolites are steroids, phenolics, essential oils, alkaloids. These are all examples of secondary and primary metabolites. The last part of the lecture is downstream processing. Downstream processing is actually the recovery and the purification of the desired product from its crude extract. For example, we did this process of extracting ethanol here and here penicillin. Now, how do we purify this ethanol, or how do we purify this penicillin, or how do we purify, let's suppose, insulin, with something we discussed in the last lecture? How do we have a pure product, clean, pure product? The technical term for this process is downstream processing. So you start with the culture broth, and you have intracellular products and extracellular products. If the product is an extracellular, you do not need to go for this step. For example, antibiotics that we discussed, they are extracellular. They are not released inside. But if you have a protein, we had been saying gene of interest, gene of interest, gene of interest all along the course. If the gene of interest is for a protein, it has to be for a protein. It's a gene. So the gene product is a protein. It will be mostly, in a majority of the cases, released or secreted inside the cell. Then we have to disrupt the cell if you want to grab our protein. For the cell disruption, we can use mechanical processes, physical process, chemical process, or enzymatic processes. Any process is fine. You just need to rupture the cell so that everything inside is released. Then we will remove the insolubles, because there will be now a number of soluble as well as insoluble stuff inside the cell. Flocculation, sedimentation, filtration, centrifugation, these are some methods. We can then, the next process is isolation. So evaporation. You can evaporate and then you can condense. You can absorb them to some substances. You can do the precipitation method, or you can enforce them, go through some filters and the membrane filtration with separate two different processes. After we have the crude product here, isolated, we have to purify it. Normally, chromatography is a technique that is used for it. Precipitation can also be used, but majority of the cases, it's chromatography and membrane filtration. Finally, when we have purified, we have to crystallize, or we can dry it, we can freeze dry it. Sterilization is very important. We have to sterilize it. And either of these processes, either you dry it, you lyophilize it. Lyophilization is actually freeze drying. So you freeze them under vacuum conditions, or you can crystallize them. And then after all these processes, you have finished the product and the final product is there. You can pack it and you can send it to a consumer market for use, to be taken as a therapeutic medicine, or as a food product, processed food product, or whatever is the type of the product. This was about today's lecture, and I will see you tomorrow in the interactive session at 3:30 p.m. So until then, take care. Allah Hafiz.